Okay? Okay, so naghangda ng sa laptop. Okay, for now, for our next chapter, okay, we are now on the microbial taxonomy and nomenclature. So, araw na i-forward din naman sa inyo. So, nyo na lang nasa basa sa baba. I'll just go directly sa aton nga discussion. Okay, so microbial taxonomy. Okay, taxonomy is an area of your biologic science that comprises three distinct but highly interrelated disciplines. You have your classifications. Ang ito di ba, may nomenclature kita or the naming na. So you have your classification, nomenclature, identification of your organism applied to all living entities. Taxonomy provides a constant mean to classify, name, and identify organism. The consistency allows the biologists worldwide to use a common label for every organism studied within the multitude of your biologic discipline. The common language that the taxonomy provide minimize confusion about the names, allowing more attention to be focused on other important scientific issues and phenomena. Taxonomy is important not only in phylogeny or the evolution of the history of your organism, but also in virtually early other biologic discipline, including your microbiology nga aman. Okay? So what your taxonomy before na that? Okay nga aman. Okay, for example, you have here or native languages. There is a Philippines. Okay? You have your Bisaya, your Ilonggo, Karaya, ba? For Bisaya, okay, the common thing na lang for naming example would be a dog. Okay, sa English, it's a dog. Okay, pero sa Ilonggo, you have your Ido. I think sa Rosno, you have Ido man. Pero sa Bisaya, we call it Iro. Okay, Iro. So, same lang gihapon, but it's for the naming na sa ilang area. Then you have there, it's just a general classification of a dog. Pero we need more to be specific, di ba? Sa dog, you have your different breeds. You have your Shih Tzu, you have Dalmatian, Doberman. Okay, that's a lot of type or a very separated species ng dogs. Na. So that's why taxonomy is important to have like a globally known term. Na ang maskin nga may mga linainas lang at tawag sa kada lain nga area, okay? But there is a collective name for it para hapos sa mga biologists or mga scientists to identify them. Na. So what is taxonomy? It is basically the science of classifying living things. Taxonomy, in simple terms, is how we name, organize, or classify organisms depending on their domain, kingdom, and so on and so forth. As the result of the advances of your molecular biology, classification or classic taxonomy has now become what it is referred to as your polyphasic taxonomy. This method of classification combines with the traditional genotypic or ang iyang composition, your phenotypic, ang like iyang pheno is ang iyang parent. So, muna, kung sa diin siya naghalin. Okay? Next is your phylogenetic or the evolution relationship into the general purpose classification. Yan. Your mga amphibians, like may mga relatives sila nga purely sa water lang, may mga relatives sila nga purely land lang. That's why do nag-crossbreed sila. That's what is your, what you call your phylogenetic evolution relationship into general purposes. At the molecular level, this process is multifaceted. Usually, your RNA or your ribosomal ribonucleic acid sequence hold the genome sequence, and epigenic, okay, it is a variation of not only caused by your nucleic acid sequence similarity or differences on your factor or kung ano biya ang ilang DNA composition. Okay? The polyphasic taxonomy approaches provided a more detailed but very complex analysis of your current classification system, not not all parameters clearly delineate each organism to the species level. In order, some characteristics may strengthen the organization of the genus, and some may be useful for the species level. For example, is we have your dinosaurs way back before. Okay? Then subong panahon, the closest relative of the dinosaur is not the Komodo dragon. Okay? Komodo dragon or the dako nga lizard, a lizard. Lizard. Okay? But a bird species. Though bird gid siya, lipat ko sa iyang name, but hindi ko ganyan malipatan iyang tsura. Okay, it's like close to an ostrich, but though madako-dako siya, and ang iyang TL is like may hooves. Okay, hindi siya ang papares sa mga bird species nga separated, or though iya ka manok, but hooves. Okay, so iya ka close nga similarities siya is iya ka baka, something like that. Okay, so comparative analysis of conservation of sequence such as the 16S or 16 strands of your DNA, 95% to 95 to 97%. Both techniques have the limitation that includes variation in cutoff values between the different species or your genera or lang genus. When using a single sequence such as your 16S rDNA, the possibility of gene transfer may also affect your genotypic classification. Finally, later gene transfer among organisms, particularly your bacteria, creates difficulty in such bacteria creates difficulty in the classification of the organism according to the phenotypic traits or biochemical traits and genotypic criteria such as your DNA, your G1 and C content. G is guanine, and C is cysteine. Content, which is a hallmark for diagnostic microbiology. Molecular methods have provided a means for identifying the historical core genomes used in the classification of species identification. But it is important to recognize the phenotypic expression such as your organisms. We will continue to be compounded by the variation of the genomes as a result of lateral gene transfer among your organisms. In addition to that, or to more advanced genotypic analysis, your chemotaxonomic method are more frequently being applied to the identification of the classification of your organism. This method includes your protein studies. Okay, aside from that, you have your fatty acid analysis man, and also ang iyang cell wall or ang outer layers ng imong microorganism. Mass spectrometry and more recently matrix-assisted laser desorption ionization or your time of flight mass spectrometry or your yung shortcut is MALDI-TOF MS use the separation and analysis of a high abundance peptide of the classification and identification of your bacteria isolates. Yun namin niya. You have your bacteria. Okay, ka-isolate siya sang isa ka bilog. Ang iya gina-measure yan is ang peptide sequence yan. Okay? So, very high, very specific or pinpoint siya kay for very, di ba gamay-gamay ito ng bacteria, imagine nyo na lang class. Okay? So, gamay-gamay ng bacteria, naka-isolate lang siya sa isa. Muna sa or yung mga advanced surface, enhanced laser desorption, ionization, time flight, or MALDI na lang, okay, captures the protein directly without the loss of a sample and decrease the sensitivity that evident in MALDI. Finally, new biosensor technology, like your Brucker, Daltonics Incorporated, mga company ni sila, Bilsilica, and your MA uses multiplex pool of your polymerase chain reaction or PCR primers that target the conservation of sequence in bacteria genomes coupled with a high precision electrospray ionization mass spectrometry to identify group organisms. The polyphasic approach is needed to use the data from your MALDI-TOF or MS in junction with the genotypic analysis and phenotypic identification of your organism will undoubtedly continue to evolve along with the changes in the populations of your organism. Okay? In diagnostic of your microbiology, okay, the classification, your nomenclature, identification of your microorganism plays as a central role in providing the accurate and timely diagnosis of your infectious diseases. A brief and detailed discussion of your major components in your taxonomy is important for a basic understanding of your bacterial identification and application in this microbiology. So first and foremost, dahil kita sa classification. Okay? Classification is a method of organizing your microorganism into groups or taxa based on a similar morphology, physiologic, and genetic traits. The hierarchical classification system consists of the following taxa designation. First and foremost, you have your domain. Okay, the domain is separated into bacteria, archaea, and eukarya. Okay, next is your kingdom. Kingdom contains several divisions. Okay, you have or phalia, most inclusive taxa. Ibig sabihin, hindi ka nag-branch off again. Next sa mga kingdom, you have your phylum. Okay, phylum contains similar classes. Okay, equivalent to the division of taxa in botany. Next is your class. Okay, and it contains the similar order, gapadalum na nagapadalum. You have your order. It contains similar families. Next is your family. It contains similar genera or your genus. Then your genus contains similar species. The species itself in the class, okay, is the specific epithet, lowercase Latin kung magsulat ka na. Okay, it is the kung specific epithet, lowercase Latin adjusted to become most exclusive taxa. Historical bacteria or prokaryotes or pre-nucleus were included in a single domain. However, with the more detailed analysis using your modern technology, this domain has now been separated into bacteria and archaea. Ibig sabihin sa una, ang bacteria, ang bacteria or prokaryotes or karyotes is isa pa lang na, pero sa modern na technology, ginto nga na. Okay? Then your bacteria, actually, so where was I? Nako to. Your archaea or your ancient bacteria. The bacteria contains the environmental prokaryotes. Your blue, green, or cyanobacteria are the heterotrophic medically relevant bacteria. The archaea are your environmental isolates that live in extreme environments such as your high salt concentrations naman. Usually, bacteria is very sensitive to salt. So, they cannot live that. Pero as ang pag-study na subong, okay, slightly modernized na, okay, they found a living organism or microorganism sa high salt concentrated areas. Next is mga jet fuel, extreme temperatures like you're close sa mga volcano or sa mga underwater volcano. Okay, yung mga microbes na nagabuhi. Next nga, third domain is your eukarya or eukaryotes. They have true nucleus. Also contains medically relevant organism including your fungi, your parasite, and severely taxonomical sublevel. Below the organism below the domain are noted previously. However, the typical application of your organism, classification, the microbiology, laboratory primarily uses the taxa beginning at the family designation. Sila, hindi po pugo Hindi po pugo. Hindi po pugo. Family, a family encompasses a group of organisms that may be contained multiple genera consist of microorganisms with a common attribute. The name of the family is formed by adding the suffix of your as ang ACEA Achaea, okay, to the root name of one of the genera called your type genus. For example, your Streptococcaceae family type is sa Streptococcus nga family. One of the exception to the rule of your microbiology is the family of Enterobacteriaceae. It is a name after the insect group of bacteria rather than the type of your species of your Escherichia coli. Okay, so bacteria is a prokaryotic type of species or strain are determined according to the guidelines published by the International Committee for the Systematic Prokaryotes. Species definition are distinguished using the DNA profiling, including a nearly complete 16 RNA sequence with less than 0 to 5% ambiguity in combination with the phenotypic traits. Type species should also be described in details using the diagnostic and comparable methods that are reproducible. Okay? Next is your genus. Okay? Genus, or kung in plural, it is genera. Okay? The next taxon contains the different species that have several important features in common. Okay? Each species within the genus differ sufficiently to maintain its status as an individual species. Okay? Placement of your species within a particular genus is based on a various genetic phenotypic characteristic shared among the species. Microorganisms do not possess the multitude of physical features exhibited by a higher organism, such as your plants, okay, and animals. For instances, they rarely leave any fossil records and they exhibit tremendous capacity in intermixed genetic material among seemingly unrelated species and genera. For this reason, confidentially established microorganism retardedly in a higher taxa beyond the genus level is difficult. Although grouping similar genera into common families and similar families into common orders is used for classification of your plants and animals. This higher taxa designation is ginatunga into division, class, and also order, and not useful for classifying bacteria, or kaya focus ita usually sa mga bacteria or mga microorganism. So these mga genus, mga padalom na is applicable usually sa biology nga subject where we study about all life. Okay? Pero kita sa microbiology lang ka focus, therefore hindi siya tama ka applicable. Next is your species. Okay, abbreviation is SP kung singular and SPP kung plural. Okay? It is the most basic of the taxonomic group and can be defined as the collection of your bacteria strains that share a common physiologic and genetic features and differ notably from the other microbial species. Occasionally, taxonomic subgroup within a species called your subspecies are recognized furthermore. Designation such as your biotype, serotype, and also your genotype may be given to a group below the subspecies level that shares a specific but relative minor characteristic. For example, your Klebsiella pneumoniae and your Klebsiella oxytoca are two distinct species within the same genus, which is your Klebsiella. But your Serratia odoferia is a biotype 2 and Treponema pallidum subspecies pallidum are example of a biotype and subspecies designation. Okay? Ibig sabihin, they are under the Klebsiella but they are in a subtype species. A biotype is considered the same species with the same genetic makeup but different or place differential physiologic characteristic. Okay, what do you mean by physiologic? It is ang iyang appearance or ang iyang physiological. Okay? Your subspecies do not display significant enough divergence to be classified as a biotype or a new species, although these subgroups may have the same taxonomic importance. Their usefulness in microbiology is subpar at best. Next is the nomenclature. Okay? Nomenclature na. Nomenclature is the naming of your microorganism according to the established rule and guidelines set forth in the International Code of Nomenclature of your bacteria. Ang tandaan nyo ni ang initial nga ni or ang ICNB. International Code of Nomenclature of your bacteria or your bacteria code pagid. You have your BC, before price. It provides the accepted labels by which organisms are universally recognized. Because the genus and species are the group of commonly used microbiologists, the discussion or rules governing your microbial nomenclature is limited to these two taxa. In this binomial or binomial or your two-name system of your nomenclature, every organism is assigned with a genus and a species or your Latin or Greek derivation. Each organism has a specific label, okay, consisting of two parts. You have the genus designated, in which the first letter is always capitalized. Wait lang, class. Okay, where was I? Okay, so each organism has a specific label consisting of two parts. The genus designation, in which the first letter is always capitalized, and the species designation, in which the first letter is always lowercase. Okay, the two components are used to simultaneously and are printed in italic or underlined kung naka-script. This is kung naka-script with underline. Okay, you have here, for example, your Streptococci includes your Streptococcus pneumoniae, Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus bovis, amongst other. Okay? See here, naka-italicisa or naka-slanting. Okay, therefore, hindi na siya need nga i-underline kung naka-script. Okay? Alternatively, the name may be abbreviation by using the uppercase from the first letter of the genus designation followed by a period, okay? And this follow a full species name in which never be abbreviated. Okay. Here, example, you have your S, pneumoniae, paras kita sa babaw, S, pyogenes, S, agalactiae, S, bovis. Ibig sabihin na the S itself or it is na sa Streptococcus. Okay. Frequently, as an informal designation, you have your Staphylococcus, you have your Streptococci and Enterococci. May be used to label particular group of your organism. These designations are not capitalized or italicized. Okay. And more information is gained regarding the organism classification and identification. A particular species may be removed to a different genus or assigned to a new genus name. The rule and criteria of these changes are beyond the scope of this chapter, but such changes are documented in the International Journal of your Systemic and Evolutionary Microbiology. Here sa rule sa aton nga taxonomy, kung ano ang iyang purpose. Okay, it established and maintains record of the key characteristic of clinically relevant microorganism. Facilitates communication among technologists, microbiologists, physician and scientists by assigning universal name to a clinically relevant microorganism. This is essential for the following. You have it established an association for a particular disease and syndromes with the specific microorganism, epidemiology and the tracking of the outbreak, accumulating knowledge regarding the management and the outcome of the disease associated with a specific microorganism. You have your establishing the patterns of the resistance to antimicrobial agent and recognition of changing your microbial resistance pattern. You have there understanding the mechanism of the antimicrobial resistance and detecting new resistance mechanism exhibited by this microorganism. Recognize new and emerging pathologic microorganism. Recognize the changes in type of infection of diseases caused by the characteristic microorganism. Okay, the best example of this rule is your COVID. Naman. Ang COVID ya class, okay, ever since or dugay na ara na ang COVID. But the type of COVID or your coronavirus nga maka-infect human is recently lang nagtuhaw dito sa China. Dito naging start nila keep track ang iyang origin, ang iyang mutation, okay, what caused it, then sang nag-infect na sa human, dito pagid yan nag-mutate ang virus into different variation pa. You have your mga delta variant, that is the most deadly subong nga panahon. You have your other variants pa sang COVID nga dasig maglaton pero the symptoms it gives you is low or ang iyang lethality rate is low. That's why you have different types of COVID na muna subong. You have your Delta, your COVID-19 nga pinakauna nga nag-infect sa human muna. Then you have your identification. Next na, okay, identification. Microbial identification is the process by which the microorganism's key features are delineated. Once those features have been established, the profile is compared with those other previously characterized microorganism. The organism can be assigned to the most appropriate taxa. Okay, this is for mga organism pa nga abi mo newly discovered mo, but since gin-characterize mo na tanan-tanan, then you refer it to your amo to international microbiology nga mga naming sang bacteria or microorganism. Na. So, that organism can be assigned to given appropriate genus and species name or nomenclature. Both are essential aspect of the role of taxonomy in microbiology and management of infectious diseases. The identification method, a wide variety of your method and criteria are used to establish microorganism identity. This method usually can be separated into two general categories. You have your genotypic and phenotypic characteristic. Your genotypic characteristic related to an organism genetic makeup, including the nature of the organism, genus, and constantly nucleic acid. Okay, see sa chapter 2 word information about microbial genetics. Your phenotypic characteristics are based on the features beyond the genetic level, including the both readily observable characteristic and features that may require your extensive analytic procedure to be detected. Example of the characteristic used as a criteria for bacterial identification and classification are provided in Table 1-1. Okay. Modern microbial taxonomy used in the combination of several methods organism through the classifying of the name and organism, although the criteria example sa dalawang sa ating table may be a concept of your microbial identification for classification purposes, and the principal practices of classification is parallel to the approaches used in diagnostic microbiology. Okay, for the identification and characterization of your microorganism encounters the clinical setting. Fortunately, because of the previous efforts and accomplishment of the microbial taxonomists, okay, or microbiologists do not have to use several burdensome classification and identification schemes to identify infectious agents. Instead, microbiologists use key phenotypic and genotypic feature in which to base their identification to provide clinically relevant information in timely manner. This should be taken to means that the identification of all clinically relevant organisms is easily and straightforward. This is also meant to imply that the microbiologists can only identify or recognize microorganisms that have already been characterized and named by a taxonomist. Indeed, the clinical microbiology laboratory is well recognized as the place where previously unknown and uncharacterized infectious agents are essentially encountered. As such, it has been an ever-increasing responsibility to the source of information and reporting for emerging etiologies of infectious diseases. So, ayan muna natin ang balta sa Table 1-1. This is the identification criteria and characteristic for the microbial classification. You have here criteria and characteristics sa piyak na side. You have your macroscopic morphology. Summarize na at least dahil siga. Okay, macroscopic. Okay, the word itself, macroscopic, the microbial growth pattern or artificial media observed when inspecting gamit lang sang aton nga eye. Okay, for example, is the size, texture, and pigmentation of your bacterial colony. So, ginagtanaw mo ni sa culture media. Okay? Next is the microscopic morphology. Okay, micro, okay, ibig sabihin, the size, shape, intercellular inclusion, cellular appendages, and arrangement of the cells when observed sa microscope. Next is the staining characteristic. Okay, the ability of the organism to produceably stain a particular color when an application of a specific dyes and reagent. Staining is used with a conjunction with the microscopic morphology of your bacteria. Dudili ang color. Used in conjunction bacteria identification. For example, you have your gram staining. Gram staining is the most common way of dyeing your bacteria. It's crucial and critical criterion for identification, differential identification. Next is the environmental requirements. Okay? It's the ability of the organism to grow at various temperatures in which the presence of oxygen and other gases and various pH level or in the presence of ions and salts such as your sodium chloride. Tama. May mga bacteria nga gabuhi lang sa mga area nga wala sang oxygen. Okay. May mga bacteria nga gabuhi sa abundant ang oxygen. Mga bacteria like example ko to sa example ta, ang mga bacteria gihapon nga gabuhi sa high temperature. Kung magdubo na ang temperature ng ginabuhian nila, they will die. Next, the resistance profile. The exhibition of the characteristic inherent resistance to a specific antibiotic, heavy metal, or toxins. Okay, for example, subong you have your methicillin resistant Staphylococcus aureus sa chapter 1 na discuss ko na. That is Staphylococcus aureus nga resistant na sa methicillin. So, ngalan niya pa lang. What happened is ang person nga infected sa Staphylococcus, okay, gatake sang bulong sa methicillin with a lower dosage. Okay, ibig sabihin, may mga napatay ng mga bacteria or Staphylococcus, okay, but not all. Okay, then ang mga nabuhi na, okay, because due to their higher resistance sa low dosage ng methicillin, okay, their resistance improve pagid. Therefore, ang gaka-develop dayon nga next nga Staphylococcus aureus nga halin man sa iya is more resistant na sa methicillin hasta nga hindi na gid kaya patyong sa methicillin maskin ano pa ka taas ang dosage. Next is the antigenic property. Okay, the profile of microorganism establishing by various serologic and immunologic method to determine the relatedness among various microbial group. Next is the subcellular properties. Molecular constituent of the cell that are typically and are particularly the taxon of the group, organism group as established by various analytic methods. For example, this includes your cell wall, the component of the cell membrane, and the enzymatic content of your microbial cell. Next, the chemotaxonomic property. Okay, sa name niya pa lang chemo. Okay, ibig sabihin excuse me. Okay, the chemical constituent of the cell, such as your structure of the nucleic acid, your fatty acid analysis, ang iyang protein profile as determined by your analytical method. Next, sa atong grouping is your genotypic. Your genotypic is natunga sa duwa. You have your DNA and there is the long or your deoxy or ribonucleic acid base consumption ratio. Your DNA composes 4 basic genes. You have your guanine, cytosine to galing, this is cytosine, adenine, and thymine. Okay, the extent of which DNA or two organism is made up of your cytosine and guanine. For example, your G1, G1C. Okay, content related to the total base content can be used as an indicator for relatedness or lack of. Therefore, for example, an organism with a G1C content of 50 is not closely related to an organism with a G1C content of 25. Okay nga aman, because ang ilang nga percentage ratio. Okay? Next is the nucleic acid or the DNA and ribonucleic acid or your RNA base sequence of the characteristic, including those determined by hybridization assay. The order of the bases along with the strand of your DNA and RNA is known as the base sequence, which extend to known as the bases to which extend to which sequence of are homogeneous or similar. Between two microorganisms can be determined directly or indirectly by various molecular methods. The degree of the similarity in the sequences may be measured in the organism relatedness, specifically in their RNA or ribosomal RNA sequence that remain stable in comparison with the genome as a whole. We have here mga tips na lang ni sa dalom, yung mga trivia, ang mudasig lang na di. Ito na lang. So, this is prepared by Sir Nyadoliete. Okay? Do summary. Okay? We have here taxonomy. It's the science of organizing and naming your living organism. Okay? How do you classify your living organism? By following this order. Okay? You have your domain, kingdom, phylum, what? Then you have your order, family, genus, and your species. Okay? In all, there are more than 8 classifications, but these 8 are most important in the hierarchy classification for you to better understand taxonomy for its mnemonics. Okay, ang mnemonics niya is Dina's kid prefer candy over green spinach, or pwede man did Katy Perry come over for a grape soda. Okay, depende na si mo kung ano ng mnemonics imo nga gamiton. Next, you have here there are only 3 domains in life, and they are your bacteria, archaea, and eukarya or eukaryotes. Number 1 and number 2, on the other hand, are prokaryotes, as stated at the start of the lesson. While number 3 is or your eukaryotes are known as eukarya, also known as your eukaryotes. So what is the main difference between the two? Okay, your eukaryote, okay, and your eukaryotes. Prokaryotes don't have nucleus. Okay, although they are cells, but they don't have nucleus. Also, no membrane-bound organelles. They are also unicellular. And so, they are taught in biology. And eukaryotes, they have what you call your nucleus, a membrane-bound organelles, and they are also multicellular. Okay, this picture below is the example of the domain of your eukarya. Okay, the organism I will use is the Homo sapiens or human. We have here Homo. We have here the kingdom Animalia or the animal kingdom. You have here the phylum Chordata. Okay, nang nagawa lang da. You have your dulaang lobster. Okay, because of sea creature gid. Next is your mammalia. Okay, ano nadula? You have nadula ang mga egg laying. Okay, therefore, they give birth. Mammalia. Next is your primates. Nadula ang ganding, beaver. Okay, nabili ng unggoy and also the ape. Then, your family of Hominidae. Okay, nadula ang monkey, nabili ng ape and ang Homo sapien kag Homo erectus. You have your genus, you have your Homo sapiens, and another Homo. Next is the species of sapiens, which is commonly known as your human. Based on this picture, the main eukarya below gets specifically represented by rabbits, crab. Ang mga muna. Explain ko na to. Diding rabbit. Why man rabbit po? Okay, specific English Homo sapiens and classification shows as we further go down. Okay, let's see your Homo sapiens classification order as we go further. Relatedness to a species closer than the other, based above your DNA testing until that our species human known as your Homo sapien. This is how taxonomy is used to identify and naming our name living organisms that are being used in scientists. Next nga term is your phylogeny. Okay, this shows us the natural relatedness between groups of your organism. I will illustrate an example picture below sa dalom. Dere. Medyo natunga lang. Okay. So ang may common ancestor, alin, ng like a single cell. Separate siya into three. You have your bacteria, dere sa piyak. Next is your nag-branch out, your eukarya and your archaea. Okay, your archaea, dire. Your eukarya, dire. You have your mga true nucleus. Okay, true nucleus, you have your protesta. You have your plantae, mga plants. Then you have your, ang mga plants na yun, natunga pa rin sa fungi and animals. Okay, this is a very simple illustration lang. Okay, this picture shows that the cell of your common ancestor and the branch out into a more complex cell such as your bacteria, archaea, and eukarya. Then those three branch out individually into new cells, organism, but illustrate states that they all have started from one cell. This shows its relatedness. Another common example of your phylogeny could be the latest COVID-19 hangamba ko, in which virus was also another new strain of your pneumonia virus, which branched out and mutated to be immune to old medical treatments. But where did it came from? Of course, came from a virus already existing, like your pneumococcal pneumonia and chlamydia pneumoniae. In essential phylogeny shows that the natural relatedness of between groups and organisms is also known as your evolutionary history of your organism. So that's all for our taxonomy. Sa dalog nga part, goals na lang ni. Sige na gani, mabigo tapos na. Okay, rules for your taxonomy and naming of your scientific simplified. Okay, very simple na gani sa babaw. For a proper naming sa mga microorganism. When writing a name of your organism, there are rules we follow in order to write it correctly, which is the follow. First is the genus must be written in the first before the species name. You have here Homo sapien. Okay, this is the genus Homo. Then ang species sapien. You have your Staphylococcus aureus, genus, species. The first letter of the genus must always be capitalized and the rest is lowercase na. Okay? You have here capital H, capital S, and capital E of Escherichia coli. Next, number 3, when typing, okay, for the name of the organism in a computer, you must always italicize na. Okay, you have here Homo sapiens, or kung handwritten, or Homo sapiens, kung type, ka-italicize. Kung handwritten, Homo sapien, or pwede man ipaslanting mo or italicize mo gid ang pagsulat. Pero kung naka-script gani class or naka-normal nga pagsulat nga patindugid, dapat may underline. You have here type, you have here Streptococcus pyogenes and Streptococcus pyogenes kung handwritten. Okay, depende na lang. So you might be asking, where do all the organisms get their name from? Usually it is named after their founder or sinong naka-discover. Example is your Theodore Escherichia, who was discovered the bacteria Escherichia coli, and they are named after the shape of their respective organism. Okay, Staphylococcus aureus, which comes from the word staphylococcus, focus, which is the shape of a cluster or a little circles when they are looked in a microscope. Or pwede man sa ilang morphology. Okay, so this ends the chapter 2. Thank you for studying Kapsudayaw. Okay, stay safe and God bless kids. So update na lang sa aton nga group page. I will upload this and also ang quizzes. Tingbon ko na lang upload. So God bless good always and take care. See you next time.